Okay, we're back. This is going to be part 2 of cellular respiration. In the first part, we talked about some broad ideas and the idea of activation energy and enzymes and how to make ATP. Now we're going to talk about the specifics, the different processes that go on in the cell that extract energy from your food and put that energy into ATP. Now, we're going to get into a little bit of detail, but not tremendous detail. There's a lot more detail. We're going to kind of have about the 10,000 foot view, maybe about the 9,000 foot view. So we're going to have a little bit of detail. What I don't want you to do is I don't want you to memorize every molecule of every step here. That's not what we're trying to accomplish in this class. In other classes, you probably will do that, you know, and if you go on to med school or vet school, you'll need to know all those different molecules. I want you to concentrate on what is in the PowerPoint. And so if I mention a molecule in the PowerPoint, then that's a molecule you need to know. But don't memorize every single step. What I want you is to understand the broad picture and how these different steps relate to each other. All right? So let's get to it. So this last part is the steps of cellular respiration. Now we mentioned ATP and the structure of ATP. We mentioned how ATP acts like a battery. It takes energy from one place and carries it to another place. So we need to charge up those ATPs. We need to take an ADP, which is an adenosine diphosphate, and we need to add another phosphate to it. Now remember, you've already got 2 phosphates, and they're repelling each other, and you're going to bring a third on. It takes a lot of energy to do this. So consequently, that bond is going to have a lot of energy in it. And when you break that bond, you release a lot of energy. Well, where are we going to get enough energy to glom that last phosphate on? We're going to get it from your food. And this is metabolism or cellular respiration. So there are 3 broad steps to cellular respiration in animals: glycolysis, then the Krebs cycle, and then the electron transport chain. So let's talk about each of these in a little bit of detail. So here's a figure from your book, and it shows how these 3 things relate to one another. The first thing you want to understand is that the products from one feed into the next one. So we start with glycolysis. The end result of glycolysis feeds into the Krebs cycle. The end results from the Krebs cycle feed into the electron transport chain. Now, each of these 3 also produce some ATP, and that's important. But what's important about the first 2, about glycolysis and the Krebs cycle, is that they ultimately feed things into the electron transport chain, and the electron transport chain is where you get a whole lot of ATP. All right? But they have to occur in this order. So we start with glycolysis. It was the first to evolve. It's very simple, and it doesn't require oxygen. This takes place just in the cytoplasm of the cell. You don't need a specialized organelle. So these are a couple of reasons why we think it evolved first. Because it is so simple, because it doesn't require organelles, you know, it can happen in prokaryotes. So it probably was the first general method for extracting energy from your food. The problem is it's not very efficient. At the end of glycolysis, you've only made 2 ATPs. That's not a lot of ATP. What that means is there's still a lot of energy locked up in the food cell, or in the food molecule, that you're wasting. And so that's the downside of glycolysis. The upside is it's very simple and you don't require oxygen. Now here's a figure from your book showing the steps to glycolysis. And again, don't worry about memorizing each of these steps for this class. Just worry about the general idea behind something like glycolysis, but any specific molecules I mentioned you need to know, such as glucose. So you'll notice that that this process always starts with glucose. And we've talked about glucose, we've talked about it's the a very important carbohydrate. And basically, that's just the way the process works, is that whatever molecules you eat, they eventually can be broken down into glucose, or at least a lot of them can be broken down into glucose. So that's kind of what digestion is, right? When you eat food, you have to break it down. Ultimately, you have to break it down into a glucose molecule, and then that can go ahead and feed into these cellular metabolism reactions. Also, whenever we talk about the number of ATPs that are yielded, it's usually per glucose molecule. That way we can compare between different molecules. So that's just a basic idea you need to understand. Now, you'll notice at the very start of glycolysis that it shows that we are using 2 ATPs. So we're taking 2 ATPs, stripping off a phosphate, which releases energy, and we're using that energy in this series of reactions. So right at the start, we're already in the hole. We haven't gained any energy. We've actually used up energy. It's cost us energy. But remember, you know, we've got this idea of activation energy, or priming the pump, that you have to put some energy in to stress bonds so that they can break, and you end up getting more energy out. And so this is an example of when we're doing that. And so basically, once you break that glucose in half, then you've got a couple of 3 carbon molecules, and then you break those molecules down. And each time you break the bonds, you release energy. And during a couple of these steps, that energy is used to stick a phosphate onto an ADP and to make an ATP. This is how we charge up an ATP. And so if you notice that we end up making 4 ATPs, all right? But we've already spent 2. So our net is just 2 ATPs per glucose molecule in glycolysis. So it's not very efficient. There's still a lot of energy tied up in these molecules. So again, you'll notice that we start by breaking a phosphate off of an ATP, which gives us some energy. And then later we take energy from that food molecule to reverse that reaction and to stick some phosphates back on to an ADP to charge up and to make ATP. Now that ATP has that energy, because that last phosphate's a very high energy molecule. It takes a lot of energy to stick it on. So now we've taken energy from the glucose molecule, and now that energy is in the ATP molecule. Relate this back to the structure of ATP. It's just a nucleotide. It's got a nitrogenous base, a 5-carbon sugar. The nucleotides that you use in DNA and RNA just have 1 phosphate, but this is ATP, adenosine triphosphate. It's got 3 phosphates. It takes a lot of energy to stick all those phosphates together because they don't want to be together, because they all got this negative charge. And so that's why these bonds have such high energy. And so, you know, glucose is a hexose. It's a 6-carbon sugar. It immediately gets cleaved into 2 identical 3-carbon sugars. And so at the end of glycolysis, you've got 2 pyruvic acid molecules. So you want to know that. You want to know pyruvic acid comes out of glycolysis. And you want to know that pyruvic acid has 3 carbons. So we start with a 6-carbon molecule, and we end up with 2 3-carbon molecules. And there's still a lot of energy in each one of those pyruvic acids. So we said that glycolysis does not require oxygen. So this can take place whether there is oxygen or there is not oxygen. If there's no oxygen present, that pyruvic acid gets converted to something called lactate, and then we're done. And lactate is not used for anything else. And so we only get a couple of ATPs out of it, and it's better than nothing. But this is not enough energy to power large organisms. And so that's why, you know, Glycolysis is fine in the early organisms on Earth. They were all small prokaryotes. Glycolysis is fine in bacteria and things, you know, again, single-celled organisms that live here on Earth now. But larger organisms require more energy, and so you're not getting enough ATP from your food if you just use glycolysis. When this occurs in the lack of oxygen, this is also called fermentation, and you're familiar with fermentation and what bacteria and yeast can do in the absence of oxygen. So this extracts energy from the food, but not a ton of energy. Where are some examples where you're going to find this? Well, anaerobic environments. So like the bottom of a pond or in a wetland or in a sealed container with yeast that doesn't allow oxygen in. Say you're fermenting, trying to make beer or something like that. So you've got a lot of things that live in these anaerobic environments because they don't need oxygen. But you don't have a lot of multicellular organisms that live there. These are all you know, single-celled organisms because they get enough energy to power that single cell, but larger organisms don't get enough energy from glycolysis. This also occurs in things like white muscle, you know, sprinter's muscle, fast twitch muscle, muscle when you initially take off running or something like that. You're using muscle that doesn't require oxygen, and so you can just get a tremendous amount of, you can get your energy from glycolysis immediately, but you can't sustain that for very long because that lactate builds up and causes other problems. Okay. So if oxygen is present, then the products from glycolysis can move to the Krebs cycle. This is also called the citric acid cycle, and I'll use those two terms interchangeably because they are the same thing. So the pyruvic acid, instead of being converted to lactate, can be used in the Krebs cycle. Now, whereas glycolysis took place in the cytoplasm of the cell, the remaining reactions are all going to take place inside the mitochondrion. So you remember the mitochondrion is the powerhouse of the cell. It's where we break down our food and extract energy. And what we're specifically talking about are these reactions. The citric acid cycle and the electron transport chain are all inside the mitochondria. So that's why the mitochondria is the site where we extract most of our energy. So if oxygen is present, those pyruvic acid molecules get converted to something called acetyl-CoA, which stands for acetyl coenzyme A. And that's just a funny name for this molecule, but you're going to see this acetyl-CoA. It shows up in a lot of biochemical reactions. And so that's what this figure is showing. It's just showing the pyruvic acid being converted to acetyl-CoA. Now, acetyl-CoA is only a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. So in this process, you lose a carbon. And so you can see, as part of this figure, carbon dioxide being, carbon being stripped off, forming carbon dioxide, and then you get rid of it. And so this is one reason why we breathe out CO2, right? You know that you breathe in oxygen, you breathe out CO2. Where does the CO2 come from? Right here. This and many other reactions I'm going to show you, but this is one spot. So that carbon dioxide you're breathing out was in a glucose molecule at some point, but then when that glucose got broken down and then converted to pyruvic acid, and the pyruvic acid got converted to acetyl-CoA, you had an extra carbon you got to get rid of. Just breathe it out. Okay, so that acetyl-CoA then can enter the Krebs cycle. And so when you broke down the glucose, you ended up with 2 acetyl-CoAs. They both go into the Krebs cycle. And so what do we mean by the Krebs cycle? What's the cycle about it? This is, again, when you get into cell biology, physiology, when you learn more about respiration, you see you've got lots of these cycles. And these cycles are basically just a series of reactions that take place one after another. So one reaction creates products that go into the next reaction, which go into the next reaction, which go into the next reaction. But eventually you come all the way back to the start. And so you've got this series of reactions that just constantly takes place as long as you can keep feeding the initial molecule into it. And so the Krebs cycle is a good example of that. We feed this acetyl-CoA into the Krebs cycle, and then you get a whole bunch of other reactions that take place. So don't memorize all these reactions, just memorize the ones that I emphasize. You'll notice as you study this Krebs cycle that, again, there are points in the cycle where we break off a carbon and get rid of it, and we form carbon dioxide. And so there's a couple spots in this cycle where we form carbon dioxide. Again, this is where, this is why you breathe out carbon dioxide. You'll notice that the acetyl-CoA comes in and it's got 2 carbons, and there are 2 places where we lose a carbon molecule. And so since it's a cycle, we want to, end up with the same number of carbons we started with. So those 2 carbons that came in with acetyl-CoA at some point have to be stripped off and you breathe them out. But in the process of all these reactions, we're building other molecules. We don't just strip off that carbon dioxide and strip off that carbon for no reason. When we break that bond, we release energy. That energy can be stuck into these other molecules. You'll also notice, if you study these reactions, that there's a couple places where we take in a water, where we need water to come in as part of the reaction. So, you have to drink water all the time. This is a big reason why you have to drink water all the time, because you need that water to participate in the Krebs cycle reactions. And so when this is all said and done, you're going to break apart that acetyl-CoA. You're going to, harvest the energy from it, but you're only going to make 1 ATP. So that's not very impressive, right? You netted a couple from glycolysis. You only get 1 out of the Krebs cycle. That's still not great. It's better than nothing. It's better than glycolysis, but it's not tremendous, right? Well, that's because we do make a whole bunch of other molecules that are going to be very important to us. So you notice that there's several spaces where we make NADH and FADH2. And you'll forgive me, I don't remember what those acronyms stand for. Nicotine, I'm afraid I don't remember what those are. But those are very common molecules. You're going to see these over and over again. And so you'll notice that we take an NAD and we add an electron to it and we get NADH. We take an FAD and we add a couple of electrons to it and we get an FADH2. Those molecules are going to feed into the electron transport chain. So those are very important molecules. At this point, we still haven't harvested their energy, but we will in the next step. And so, excuse me, like I said, the way we build those is we take electrons from the food molecule and we give them to NAD or FAD. And when you get those electrons, again, there's also protons laying around, and so maybe you'll grab a proton to balance out the charge. These are details that you would get in other classes. The most important thing is that you strip an electron from your food molecule and you give it to these molecules. That takes a lot of energy, so consequently those electrons have a lot of energy. So these are high energy electrons that you add to NAD or FAD. And so we're going to use those high energy electrons in the next step, which is the electron transport chain. So let's talk about that. So you've got a whole bunch of electrons that came from your food. Those electrons have a lot of energy. Those electrons are in NADH and FADH2. Well, one thing we've talked about before is how oxygen really, really wants electrons, right? It wants to use those electrons to fill out its outer shells. And since oxygen really wants electrons, let's give them these electrons that we just scrubbed from our food molecules. But we're not going to hand them over directly. We're going to kind of pass them from enzyme to enzyme, and then eventually oxygen will get them. Well, that's why it's called the electron transport chain. We've got a chain of enzymes that sit next to each other, and they transport an electron from one to the other and eventually to oxygen. So where did these electrons start with? They started in your food molecule. They started way back in the glucose originally that was broken down in glycolysis, right? And those electrons came with the acetyl-CoA into the Krebs cycle. Then in the Krebs cycle, those electrons got pulled off and stuck on and made to make NADH and FADH2. Now we're going to take those electrons from NADH and FADH2. We're going to pass them. I'm going to pass them to this enzyme. This enzyme is going to pass to that enzyme. This is going to pass to that enzyme. We're going to pass them down the chain and then eventually hand them off to oxygen. Well, first off, you see why this only takes place when oxygen is present in an aerobic environment. If there's no oxygen, then there's nowhere to pass these electrons. And so this whole thing breaks down. This is why you have to constantly breathe in oxygen, because you need that oxygen to be what's known as the terminal electron acceptor, the molecule that's going to grab these electrons. If there's nothing there to grab those electrons, then the electrons just pile up. And so, you know, this enzyme grabs an electron and it wants to pass it to the next enzyme, but that enzyme already has an electron. And that next enzyme already has an electron. And so you can't pass electrons because they're not going anywhere if you don't have oxygen. As long as you keep breathing in oxygen, then you can keep grabbing these electrons and the electron transport chain can keep functioning. And why is oxygen so important to this? Because of the properties of oxygen. Remember, it really, really wants a couple of electrons that'll help it fill out its outer shell. And so because oxygen is there sucking up and greedily grabbing up electrons, well, we can take advantage of that and drive the electron transport chain. Okay, so remember, these last two processes, the Krebs cycle and the electron transport chain, they both take place inside the mitochondria, whereas glycolysis takes place out in the cytoplasm. So the Krebs cycle is in the inner part, the very inside of the mitochondrion. So if you look at this mitochondria, it's got a double membrane. It's got a membrane inside another membrane, right? It's unique. Not all organelles have this. So the very inside portion is where the Krebs cycle takes place. The electron transport chain takes place along that inner membrane. The outer membrane, you know, defines the outside of the mitochondrion. The inner membrane is where electron transport takes place. So let's zoom in on that membrane. And so we've just zoomed in on that inner membrane. And so in this example, in this picture, you can see the outer membrane and the inner membrane. Between these two is an area called the intermembrane space. Now remember, again, what a membrane is, right? It's a phospholipid bilayer with lots of protein stuck in it. This is no different. This is the same kind of membrane. And so that inner membrane is a phospholipid bilayer with lots of proteins stuck in it. But those proteins in this example, those are the enzymes that pass electrons. That's the kind of protein they are. And so that's what they're trying to show you here. They're trying to show you the NADH donating an electron. So where did it get that electron from? It got it from the Krebs cycle. So it donates that electron to the first protein, the first enzyme in the electron transport chain. Then that enzyme passes it to the next enzyme, which passes it to the next enzyme. And then eventually it passes it to an oxygen. So that's why it's a chain. It's a chain that transports electrons. Well, again, what drives this? Why do those electrons keep moving? As long as you keep feeding oxygen, that oxygen really, really wants electrons. And so once an oxygen molecule grabs electrons, then it's cool, then it splits. But you've got another oxygen molecule coming in, and another, as long as you keep breathing. And so instead of handing those electrons directly to oxygen, we pass it from enzyme to enzyme. But in doing so, we can make those enzymes do a little work. And so here in this example, you can see where that oxygen is grabbing that electron, and that's where the electron will stay. And then when it does this, it can react with some protons, and it can make a water molecule, and it's happy. And so again, that's why we call oxygen the terminal electron acceptor, or the last electron acceptor. It's where those electrons end up. So remember that the whole point of all of this is to take energy from your food and charge up some ATPs. But we haven't done we netted 2 ATPs in glycolysis. We only got 1 per molecule in the Krebs cycle. We still have not made a lot of ATP, but we're going to. Because, like I said, as that electron gets passed from enzyme to enzyme, remember that it's a high energy electron. That electron has a lot of energy. And so as that enzyme passes that high energy electron, it can do some work. It can use that energy to change its shape a little bit. And so when that electron gets passed from this enzyme to that enzyme, the energy in that electron causes the protein to change shape. As it changes shape, that causes the enzyme to pump a proton across the inner membrane into the inner membrane space. So that's the kind of work these enzymes are doing. As they pass the electron, they get enough energy to pump protons. And so how come they're able to pump protons? Because they're changing shape, right? Proteins, it's all about changing shape. It's all about changing shape. They wouldn't just do this on their own, but now that they have this high energy electron, they can use that energy to change shape and to pump protons. Now remember, a proton is just a hydrogen that has lost its electron. And these are laying everywhere. So it's a hydrogen ion. And so you remember, we measured pH, right? We measured the concentration of these hydrogen ions because they're so common and they're so important and they're so reactive, and you find them in so many situations. And this is a perfect example that we've got these protons just laying around everywhere inside the mitochondrion. And so when this enzyme passes the electron, it just grabs a proton and just pumps it across the membrane. And so again, if you look at this and study this figure, this is what we're trying to show you. And so as that electron is passed from enzyme to enzyme, each one of them pumps protons into that inner membrane space. Now, consequently, there are a lot of protons in that inner membrane space. There's a high concentration of protons in this space, right? So we are pumping protons against their concentration gradient. We're moving these hydrogen ions, these protons, from a low concentration to a high concentration. And you remember that doesn't happen spontaneously. That requires energy. How are you going to do this if you need to use energy? Well, we just got energy. We've got a high energy electron. And so this is how we use energy to pump these protons where they don't want to go, against their concentration gradient. So this inner membrane space has a whole heck of a lot of protons in it. So we've got a high concentration of protons on one side of a membrane. Well, what happens when you have a high concentration of something on one side of a membrane? They want to diffuse down their concentration gradient. They want to go back to the area of low concentration, right? Just like anything else. This is why we made such a big deal about diffusion. Here's an example of where diffusion is used to drive a cellular process. So you've got a high concentration of protons in the inner membrane space. To get more protons in there, it's not going to happen spontaneously. We had to use energy, but we just did use energy. We got the energy from this enzyme to pump those protons. There's low concentration of protons in the middle part of the mitochondrion. And so, I already gave this away, but what will happen when we have a high concentration on one side of a membrane and a low concentration on the other? Well, the molecules will diffuse from high concentration to low concentration. And that's what these protons want to do. So you've got that high concentration in the inner membrane space. It wants to diffuse back to the other side of that membrane. But just like before, it can't just go through the membrane. It's got to find a hole. And so we've got special enzymes that make a hole for those protons to diffuse back. And those enzymes are called ATP synthase. And so those protons diffuse back through the ATP synthase. Now this is an enzyme that it's not just there to make a hole for protons, it's there to take an ADP and a phosphate and glom them together to make an ATP. Now you remember we said that takes a lot of energy to do that. Where's it getting that energy? Because that proton is diffusing through, that causes ATP synthase to change shape, which causes it to catalyze this reaction. And so now we have finally made some ATP, right? It took us a few steps, but we did it through a kind of a, it's kind of a a little bit of hillbilly engineering here almost. But then again, it's also pretty cool how this works out. So the energy from your food is in that high energy electron. As that gets passed from enzyme to enzyme, it pumps protons against their concentration gradient. They really want to diffuse back. They diffuse back through ATP synthase, and that causes that enzyme to change shape, which allows it to make an ATP. And so in this way, we can make a lot of ATPs because we have a lot of these electrons. So again, adding that third phosphate takes a lot of energy, right? Those negative charges all repel each other. And so if you are able to get that last phosphate close enough to where it'll bind to the other ones, then that chemical bond has a lot of energy in it. How can we do this? It takes a lot of energy. Where do we get that energy? Our food has a lot of high energy electrons. And those high energy electrons are stripped from the food in the Krebs cycle, and then they're given to the electron transport chain. The electron transport chain passes those electrons from one enzyme to another if oxygen's present, and then oxygen runs off with the electrons. In the process of passing those electrons, you pump protons across their concentration gradient. Those protons are a real high concentration in that inner membrane space. They want to diffuse back, and they will diffuse back without an extra input of energy. But as they diffuse back, they drive ATP synthase, which makes a bunch of ATPs. And so in the end, we're using oxygen to phosphorylate ATP, meaning we're adding a phosphate, that's phosphorylation. And so since we're using oxygen to phosphorylate this ATP, it's called oxidative phosphorylation. We can also call this aerobic metabolism or aerobic respiration. The important part is that you got to have oxygen to drive the electron transport chain. And so the enzyme here, called ATP synthase, adds a phosphate to an ADP to make ATP. What gives it the energy or the ability to do this? As protons diffuse back through it. And those protons really want to diffuse back through it because, you know, they're currently at a high concentration. They want to diffuse back to a low concentration. But in the process, that changes the shape of the enzyme and allows it to phosphorylate this ADP and make an ATP. Okay. Now remember that we did make a few ATPs earlier. In glycolysis, we made a couple. And in the Krebs cycle, we made one per molecule. Those are made in a different way. You're not pumping protons there. That's a different enzyme, and it's using the energy in a different way, but it's basically, ultimately, it's the same process. Ultimately, the energy causes that enzyme to change shape, which allows it to bring an ADP and a phosphate together. But it's just a different kind of reaction, a different kind of enzyme. In the electron transport chain is where we're pumping these protons and then using their diffusion to phosphorylate the ADP. And so that's it. So we took energy from our food and we transferred that energy to ATP. And so now that ATP can go to 1,000, 100,000 other reactions in your cells which need ATP. So the rest of the semester we're going to be talking about these things, and we're going to talk about this uses ATP and that uses ATP, but this is how we can make those ATPs. Glycolysis gave us a couple. Citric acid only gave us one per molecule, but the citric acid cycle gave us a whole bunch of molecules with high energy electrons. Those high energy electrons went to the electron transport chain. As the electrons get passed from enzyme to enzyme, those enzymes pump protons against their concentration gradient. Eventually oxygen takes those electrons, which have now lost their energy, and oxygen makes a water and just splits. Those protons are really concentrated. They want to diffuse back. As they diffuse back, they drive the synthesis of ATP. Now, each time, you know, we're not making new energy, we're just converting energy from one form to another. The first law of thermodynamics says that you don't make energy, you just convert energy. The second law of thermodynamics says each time you do this, you lose a little bit of energy to heat. So every time we break a bond and we harvest that energy and put it into a different bond and use it to change the shape of a protein or use it to pump a proton or whatever, we lose a little bit of that energy to heat. That's why your body stays warm, right? And that's why every one of these reactions we lose a little bit of energy, and that's why, you know, you only have so many trophic levels, because eventually there's, you know, all that energy is going to turn into heat. There's not enough energy left for other living things. Okay. So, you know, take a good look at this figure in your book. Make sure you understand the relationship of each one of these processes to the others. Don't memorize each step, just memorize the ones that I emphasized. And so now let's wrap up by just talking about some of the consequences or some other interesting facts about cellular respiration. For example, what if you had a molecule that bound to a protein in the electron transport chain? And once it bound to that protein, it locked it up. And so now that protein can't pass electrons, right? So if that happens, now electrons can't flow. You know, this enzyme wants to pass an electron, but that next one in the chain has got something else bound to it. Well, if that happens, electrons don't flow, protons don't get pumped. If protons don't get pumped, ATP doesn't get built. And so then the organism is going to run out, you know, of energy. It's going to run out of ATP very quickly. Well, that's what a lot of poisons do. For example, cyanide or carbon monoxide. You know these things are bad for you. Well, this is why they're bad for you. And you know these things can kill you pretty quickly. And so you can see how important it is that these reactions keep taking place. If you stop electron transport for just a very short time, you run out of energy, you can't supply energy to your brain, and you're going to die. This is why you have to breathe all the time. If you stop breathing, you stop the electron transport chain. That's a problem. But things like cyanide and carbon monoxide stop it by binding to proteins in the electron transport. Okay, the last thing we need to talk about very briefly, what about other sources of food? You remember that all this started with glucose. And so if you eat carbohydrates, you can break those carbohydrates down into glucose, and it goes right into this whole system, right? But you eat more than just glucose, or more than carbohydrates, right? You eat lipids, you eat protein, and you get energy from those molecules as well. So where do they fit into this whole process? Well, again, there's a lot more complexity to this than what I'm going to give you, but I'm just going to kind of give you a brief overview. So like I said, the previous example always starts with glucose, right? So where do these other molecules fit in? Well, let's look at the structure of something like a lipid. A lipid is a glycerol plus 3 fatty acids. You remember that? And so when your body digests lipids, it breaks off that glycerol. Well, glycerol can feed right into glycolysis. And so when you digest a fat, you break off that glycerol can feed into glycolysis, and you can then have it, once it gets into glycolysis, then it can get broken down, get some ATP, then it can go to the Krebs cycle, then it can go to the electron transport chain. And so once that glycerol gets into glycolysis, then it can feed right through the rest of the processes. And so somewhere, I don't know exactly where, this is where in other classes you'll get into these details, but somewhere in glycolysis, that glycerol can enter these reactions. And from that point on, it can go through the rest of the reactions. And so in that way, you can break that glycerol down and extract its energy. Now, when you break that lipid apart, you also end up with 3 fatty acids. And so those fatty acids can get broken down into a bunch of acetyl-CoAs, acetyl coenzyme A's. You remember that acetyl coenzyme A feeds right into the Krebs cycle, and then the results from that feed right into the electron transport. Well, you remember that these fatty acids are big, long hydrocarbons. There's lots and lots and lots of carbons in there. Acetyl-CoA is only 2 carbons. So you can break that fatty acid into a whole bunch of these 2 carbon molecules. And so from one fatty acid, you can get a lot of acetyl-CoA, all of which goes in to the Krebs cycle, all of which makes a lot of ATP. That's why fats have so much energy. That's why you get so much energy from fats, because they can feed into these processes and extract a lot of that energy. And so for every fat, you've got 3 of those hydrocarbon tails. You can make a tremendous amount of acetyl-CoA. And so that fatty acid gets broken down 2 carbons at a time, feeds into the Krebs cycle. There's a ton of these, and so you can just get a ton of ATP once it goes into the Krebs cycle, and then electron transport. Okay, what about proteins? You also get energy from proteins. Proteins are made of amino acids. And so the first step of protein digestion is to break that protein down into those individual amino acids. Then the amino acids themselves get broken down. You remember an amino acid has the carbon skeleton, or the R group, and an amine group, and the nitrogen group. And so when you digest an amino acid, you break that amine off the carbon skeleton. And so now you've got a carbon skeleton, and you've got an amine and a nitrogen group. Well, that carbon skeleton can get broken down further and gets, again, fed into the Krebs cycle. And once it goes into the Krebs cycle, then it gets broken down, and you can strip off the electrons, and you can make ATP and send the electrons to the electron transport. All the other steps take place to extract energy from that carbon skeleton. Now, that amine group, that nitrogen you broke off, that is not something you can digest. That is not, you're not going to get energy from that. That is, makes ammonia, and ammonia is toxic. And so that's something you've got to get rid of. One reason ammonia is toxic is that it actually binds to ketoglutaric acid, which is part of the Krebs cycle. And so that ammonia can bind to this molecule, shut that molecule down, and it can shut the whole Krebs cycle down. So that's one reason why ammonia is toxic. Well, you're getting a lot of nitrogen from these amino acids, you're making a lot of ammonia, so you need to get rid of it. And so this is, we'll talk about this later when we talk about the excretory system and kidneys, but this is all animals need to get rid of their excess ammonia from the digestion of proteins. Now, ammonia is really soluble in water. And so for things like fish, they can just get rid of ammonia easily. They're in water, the ammonia dissolves in water, and they just excrete it, mostly through their gills. However, terrestrial animals usually need to process the ammonia into something that they can then excrete in the urine. And so, for example, mammals like us, we produce urea. Other organisms, amphibians, also produce urea, which is a bigger molecule that contains the nitrogen. Whereas ammonia is toxic to us, urea is not toxic. And so this is how we can store all this extra ammonia in something that's not going to be toxic to us, and then we can excrete it out of the urine. Other organisms, insects, reptiles, birds, they all produce uric acid, which is similar to urea. It's just another big molecule where you can store this excess nitrogen and then excrete it in the urine. Okay. So, we covered a lot here. Let's take a step back. What exactly did we talk about? We talked about how food molecules are broken down and their bonds are broken, and we extract that energy. And then we take the energy in those bonds and we transfer it to ATP. And then that ATP can be used in all kinds of other reactions. So there are a ton of details when you dig into this, right? So this is just an introduction to the process. That's why med school takes forever, right? Med school, vet school, you're going to learn a lot of these details, because understanding these details helps to explain a lot of sickness and disorders. Okay, well, take a look at all that, study the figures, let me know if you've got any questions, and I will talk to you later. Have a good one.